The overall goal of this procedure is to monitor protein resolubilization and refolding in vivo, in near real time via fluorescence microscopy and enzymatic assays. This is accomplished by first transforming cells with a plasmid containing firefly luciferase fused to green fluorescent protein, or FFL-GFP. The second step is to induce expression of the FFL-GFP protein, followed by cyclohexamide treatment to halt production of the protein. Next, measure luciferase activity before and for a period of time after thermal denaturation. The final step is to visualize cells to determine solubility of the FFL-GFP protein. Ultimately, fluorescence microscopy and enzymatic assays are used to quantify the efficiency of reactivation of a model unfolded enzyme in multiple strains.
The main advantage of this technique over existing methods like endpoint analysis is that you can correlate activity of the protein with aggregation status over time to determine the relationship between these two phenomena. This method can help answer key questions in the protein homeostasis field, such as how specific chaperones and co-chaperones contribute to disaggregation and refolding of a model protein, and how these two activities in the cell are dependent on one another.
The firefly luciferase green fluorescent protein fusion protein, FFL-GFP, is expressed from this plasmid under the control of the MET25 methionine repressible promoter.